mode.
Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Tumor Size Based Measurements as Endpoints in Cancer Clinical Trials. My name is Edward and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future download. A big thanks to IDDI who developed the content for this webinar. International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologic, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Raleigh, and San Francisco. And at this point, I'd like to introduce to you our speakers for today's webinar. Dr. Tomasz Bursakowski has been working as a research professor of biostatistics and bioinformatics at the Center for Statistics of Hasselt University since 2002. He held visiting professorship positions at the Karolinska Institute, Warsaw University of Technology, and Medical University of Białystok, and has been involved in interdisciplinary research in numerous biomedical studies and clinical trials. Dr. Everardo Saad has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo, and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. And without further ado, I'd like to hand the mic over to Dr. Saad.
Thank you, Edward. Thank you very much. So hello to all of you. Thank you for attending this webinar. I hope you can all hear me well. So the plan today for Tomasz and myself is to provide you with an overview, at least our view of tumor size-based measurements as endpoints in clinical trials. So we'll do this first by providing you with an overview of the role of response assessment in oncology in general and in clinical trials in particular. Then I will review briefly the traditional definition of response rates and some novel endpoints based on tumor measurements rather than response rates. Then I will hand it over to Tomasz, who will present some results of studies that are still ongoing, still unpublished, but with some available results on modeling tumor size changes. And he'll try to answer the question, is death a response surrogate for overall survival in, at least in some solid tumors? And then in the very end, I'll come back and try to provide some tentative conclusions to our presentation. So let me start in a very simple way. What are the therapeutic objectives in oncology? What do patients expect from treatment, and what do physicians want to accomplish with treatment? Well, very simply, I think it's fair to say that cure would be the ideal goal whenever possible. Unfortunately, this is not possible, especially in the metastatic setting for many patients. So extending survival is clearly high on the list of priorities, and then maintaining or improving quality of life is also of high importance as general therapeutic objectives. So if we think about the ideal endpoint, it should be able to capture all the clinically relevant events. It should be easy to measure. It should provide for little opportunity for ascertainment bias. It should be observed as early as possible and in as many patients as possible in terms of power. Therefore, it should be statistically sensitive. So the question is, does the ideal endpoint exist? We think not yet. So if we look at this very simple depiction of pros and cons, so to speak, of several commonly used endpoints. For example, overall survival is easy to measure, of course. It has very little potential for bias, and it's arguably the most clinically relevant endpoint other than cure. But it suffers from some drawbacks such as the time needed to measure survival and therefore the number of events, and as a consequence the statistical power is often suboptimal when overall survival is used as primary endpoint. Quality of life has many other problems, such as the difficulty of measuring, exactly defining what quality of life is. There is potential for bias from many sources. The statistical power is, of course, depending on what domain is used, but usually it's low. It is clinically relevant, but it's far from being the ideal endpoint. And likewise, time to progression or PFS or response rate are also far from being ideal endpoints. Unfortunately, we don't have biomarkers for most clinical settings in oncology, but ideally we should have biomarkers that are easy to measure. They can be measured in most patients with little potential for bias, great statistical power. But again, we might lack the clinical relevance for the biomarker as an ideal endpoint. So we still don't have the ideal endpoint. We struggle with this in oncology. And despite that, we have a certain hierarchy of endpoints according to which overall survival sits at the top, followed by quality of life, and then all the others endpoints. Some of them are often used as primary endpoints depending on the setting. And among these other endpoints, objective response rate, of course, sits there. It's often used as primary endpoint in phase 2 trials, more often as a secondary endpoint in phase 3 trials. Now, tumor response presents somewhat of a paradox. If we think of it as a treatment goal, it's different from when we think of it as a metric to really assess the treatment benefit in individual patients. So if we think about a treatment goal, it's clear that we want to extend survival and improve quality of life, as I said, and that tumor response per se is not something we really pursue with much effort. Of course, tumor response may be associated with symptom control in some situations, but not always. So as a treatment goal per se, tumor response is not something we value so much. However, for the individual patient, of course, survival time has no meaning, because in one single patient you can never tell what would be their survival with another treatment. So you cannot use survival to actually assess the benefit of treatment in one single patient. Likewise, quality of life is not a very good indicator of treatment benefit, because it depends on treatment, but it also depends on other things that go on during anti-cancer therapy. And so the association or the causal association between treatment and benefit in quality of life is not always very clear. And this is where the paradox comes in. Tumor response is the best indicator of treatment benefit in individual patients. So this is what I'm trying to call a paradox here. Something that is not very good as a treatment goal is tremendously useful and important in managing individual patients. Now, the discussion about the tumor measurements and response rates, of course, makes us think of these metrics as surrogate endpoints. Essentially because we still consider that survival is the most important metric in oncology. But it's clear that we need surrogate endpoints mostly to reduce the development time because surrogates can be often observed earlier and therefore we have more statistical power because they are more commonly observed. In other words, more patients will have the events of interest for surrogate endpoints than for other endpoints such as survival, and therefore this is more sensitive to the treatment effects. So a surrogate endpoint, as you know, is a biomarker or endpoint that is intended to substitute for a clinical endpoint. And an endpoint that is merely correlated to a clinical endpoint may not necessarily be a good surrogate for it, and we'll see more of that later in this webinar. Now, a surrogate endpoint is expected to predict the treatment effect on the clinical endpoint, and of course this may be benefit, harm, or neither. So a surrogate endpoint has to predict the treatment effect on the final endpoint. In this case, response should be able to predict the treatment effect on response should be able to predict the treatment effect on survival. Now, tumor size assessment has been very important in oncology since the beginning because it has always been clear that a shrinking tumor indicates effect from treatment. Other than very rare anecdotal reports of spontaneous regressions in very few tumor types, whenever a tumor is shrinking, it's because some kind of treatment is having its effect, and vice versa. A growing tumor almost always indicates lack of effect from treatment. There may be now some exceptions when it comes to immunotherapy, at least for some time, when we look at so-called pseudo-progressions. But I would say these are exceptions to the rule. Conversely, a stable tumor doesn't say much because it can be from natural history of the disease or it can be from treatment itself. So the fact that tumor shrinkage has always been seen as an indicator of treatment benefit, at least in individual patients, led to the use of tumor size assessments in clinical trials since the beginning. There was a time when there were discussions about unidimensional versus bidimensional measurements. At some point, the World Health Organization criteria were established in 1979, and so bidimensional measurements became standard. There were some differences in criteria that were proposed. For example, the Southwest Oncology Group criteria, which was less stringent for declaring progressions. And at some point, since 2001 at least, we've been using so-called RECIST criteria that are again based on unidimensional measurements, so they aim at simplifying response assessment. Over the years, there have been variations of RECIST for various disease types, for example, for hepatocellular carcinoma, for neuro-oncology, and more recently, the RECIST group has published some novel guidelines more specific to immunotherapy. Now, in parallel, there has been interest in other types of assessment that are not based on plain, so to speak, assessment of tumors, but rather on volumetric measurements based on imaging studies such as CTs, and also functional assessment, either alone or combined with morphologic assessment, has been of interest. But it's true that these types of assessments have not been implemented widely in clinical trials in oncology. So nowadays, we're still mostly using RECIST or some variations of it for most of the clinical trials in which tumor size is assessed. So in consequence, we always talk about the response rate, and essentially, very simply put, what we do is to have tumor lesions that are seen on valid imaging studies measured at baseline, RECIST will specify a maximum number of lesions per organ or per patient, and then the tumor dimensions along treatment are compared with those obtained at baseline, and then according to the best response from a patient is classified as having complete or partial response or stable or progressive disease. And what we call the response rate, of course, is the sum of the proportion of patients achieving CR plus PR, and most of the times as long as this is confirmed at least 4 weeks later. Now, it so happens that response is a continuous variable in nature. So we transform this into a binary variable by speaking of a response rate, but each patient has a certain degree of tumor shrinkage, and this becomes obvious when we look at waterfall plots such as this one. Now, one caveat of these plots is that we are looking mostly or most of the times at target lesions, and so sometimes people indicate by color coding the category of responses for each patient. So not all patients achieving the RECIST threshold for target lesion reduction will have partial response, because they may have had a different behavior in their non-target lesions or even the appearance of new lesions. So that's why sometimes color coding is used to indicate partial responses and differentiate them from other categories of responses. So not only is response a continuous variable, but it can also be measured over time. And that's what is depicted typically as these, some people call these spider plots, some people call them spaghetti plots, but that's what these plots try to depict. In individual patients, the tumor size over time in relation to baseline, and then you can have an idea for individual patients what's happening with their tumor burden. So given that response is continuous and can be assessed over time, recently there has been a lot of interest in novel ways of assessing the response to therapy. And the two ones that are more often talked about are the early tumor shrinkage and the depth of response. Early tumor shrinkage is a dichotomous outcome according to which the sum of the longest diameters at a certain time point, more typically at 6 or 8 weeks, is compared with baseline, and if there has been a decrease of at least 20%, this characterizes early tumor shrinkage. Otherwise there is no tumor shrinkage, and so in a way this is a variation of a response rate in the sense that it's dichotomous, and the attempt is to assess benefit to therapy a bit earlier than would be done if you had to wait for the response in each patient. So by assessing at a certain landmark after start of therapy, it's hoped that decisions can be made sooner. Now, depth of response is different because it's a continuous variable, and it measures the percentage of tumor shrinkage observed at the nadir defined as the lower tumor burden in comparison with baseline. So in other words, this is a relative change from baseline. So people have been doing some research. There have been many papers recently about this. And the question, of course, the questions are, first, could any of these new metrics be surrogates for OS? In other words, could they be used for clinical trials as surrogates for OS? The second question is, could they be better surrogates than response rate? And then this could be looked at both at the patient level or at the trial level. So the question is, by using one of these metrics rather than using response rates, do we gain anything? Now, we're not showing here because most of the talk from now on will focus on what we call the trial level association, but we have done some studies still to be published where we have the impression that none of these novel endpoints performs better at the individual level than the good old response rate. So at this point it's not clear whether we should use any of these to substitute for decisions in clinical practice, but it appears that all of them are more or less measuring the same thing, and all of them are good indicators of treatment benefit in individual patients. So the results that have been published so far in terms of the association between these new metrics and OS have typically focused on what we call the individual level of association between achieving these endpoints and achieving good survival. For example, on the Kaplan-Meier on the top shows patients with and without early tumor shrinkage. In this case, the cutoff was 10% and not 20%, but it's clear that patients who achieve early tumor shrinkage live longer than patients who do not. Likewise, at the bottom, the table shows that according to increasing quantiles of tumor shrinkage, the median survival also increases in a statistically significant way. In other words, there are associations between both early tumor shrinkage and depth of response and overall survival. Now, the surrogate validation requires one step further. In addition to the individual level surrogacy, which is what is more or less depicted in the previous slide, there needs to be an association between the treatment effect on the tentative surrogate endpoint and the treatment effect on the final endpoint. In other words, the clinical endpoint needs to predict for the treatment effect on the final endpoint. And this is where I will hand the presentation over to Tomasz, who will continue with data on the trial level association. Tomasz?
Yes, thanks, Everardo. Indeed, so what Everardo just mentioned is that if we talk about surrogacy, we often aim at evaluating treatment effect earlier by using the surrogate, the clinical endpoint. So in this respect, we want to predict, and this is the definition of the biomarkers definition working group, that the effect of treatment on a surrogate endpoint must be reasonably likely to predict clinical benefit. Thus, we need a prediction model which would allow us to evaluate the precision of the prediction. And in general, usually we do it by looking at the meta-analysis. So we use multiple trials in which the surrogate endpoint and the clinical endpoint was measured, and then we look at the association between the treatment effects evaluated in these historical trials. And if the association is tight, and this can be measured by, for instance, the coefficient of determination or the correlation coefficient between these pairs of points, then well, if this association is strong, then it means that, well, using a linear regression, for instance, as here, you could build a prediction model which would give you relatively precise predictions of the effect on the clinical endpoint from the effect on the surrogate. On the other hand, if the R-squared or correlation coefficient is not high, well, then it would mean that, well, there may be still some association, but it's not strong enough to give reasonable predictions. So this is the trial level surrogacy which Everardo mentioned, and that's what we will be focusing on now. So I want to go back to some historical exercises that we have conducted. This is the example of evaluation of the tumor response as a surrogate for survival in advanced colorectal cancer. In this analysis, we used 27 trials with about 4,000 patients. And in these trials, 5-FU bolus was compared with some modifications with leucovorin, methotrexate, and so on. So in these trials, in the meta-analysis, you can show that with experimental regimes, so with the modified 5-FU treatment, the response rate basically doubles. And that's obviously statistically significant. There's also small but statistically significant effect on survival, which is about 10% reduction in the hazard for overall survival. So the question is whether indeed, well, there seems that, there's an effect on response, so there seems to be also effect on survival. So the question is whether we can substitute response for survival. So this is the picture showing the effects on tumor response, so log odds ratios for tumor response and log hazard ratios for survival. And as you can see, this is a plot which indicates that there's quite a bit of spread around the regression line. This can be measured by the coefficient of determination, which is equal to 0.44, which essentially means that it's about 40% explanations of the variability in the effects on survival, and this 44% variability can be explained by the variability in the effects on the tumor response. But arguably, this association is not very much strong. What one could do is also look at the 4 categories response, meaning that we could look at the progressive disease, stable disease, partial response, and complete response. We could also estimate the treatment effect on such a response using proportional odds model. And here we have the results. Again, this is the log odds ratio for 4 category tumor response and log hazard ratio on survival. And again, what we see is quite a huge spread of these points around the regression line, leading to R² equal to 0.15, meaning that there's really a very weak association between the treatment effects on these 2 endpoints. So we can say that, you know, there's a less than half of the variability in treatment effects upon survival, which can be explained by the variability in treatment effects upon response. So clearly, if we think about the prediction of the treatment effect on survival, response is not a valid surrogate at the trial level. Now, this is advanced colorectal cancer. One could ask the question, how about other cancer types? And this was actually another analysis which we have conducted. This was advanced breast cancer, 11 trials with, again, about 4,000 patients, comparing taxanes with anthracyclines. And here is, again, the plot of the effect on tumor response, so log odds ratio on the, let's say, logarithmic scale, and log hazard ratio. And we see a similar picture as in advanced colorectal cancer, pretty large spread of these points around the regression line, R² equal to 0.32. If we look at disease control, we see, so this is a little bit different endpoint, so it is stable disease plus partial response plus complete response. We see that also for disease control, the association is not much better. So also in this case, in advanced breast cancer, we can say that the prediction of treatment effect on the surrogate is poor. Less than 1/3 of the variability in treatment effects upon survival can be explained by the variability in treatment effects upon response, or well, tumor binary response or disease control. And therefore, in this case, neither response, tumor response, nor disease control is a valid surrogate at the trial level. Well, then the question is, okay, well, these endpoints are categorical or binary in nature, so obviously there is a reduction in information which they carry, and all of them are based on the tumor size. So the question is, how about using the continuous tumor size measurements? Eduardo mentioned depth of response, which is based on assessment of the relative change versus baseline. So this is a continuous endpoint. So the hope might be that this actually can be more sensitive than dichotomized or categorized endpoints. So this might perhaps be a better candidate for an endpoint for a surrogate. So we have undertaken a research using the ARCAD database. ARCAD is an initiative which collects data from clinical trials, and we have used the trials in, again, advanced colorectal cancer. I want to make a disclaimer. This is still work in progress, so the results can be seen as tentative. There may be some changes, and there will be some changes because there are some things we may want to investigate and change in the current analysis. So these conclusions should be treated as tentative. But what we have done is we have defined 3 subsets of trials. One subset, you compared only chemotherapy-based regimens. There were 8 studies, some of them multi-arm, so we defined 18 comparisons, 18 independent comparisons based on these studies, totaling about 5,000 patients. We also look at the trials which involved antiangiogenic agents. These were 4 studies leading to 11 comparisons for about 4,000 patients. And then we looked at the trials which involved anti-EGFR agents, 6 studies leading to 8 comparisons, about 3,000 patients. Briefly, this is a table showing different comparisons for chemotherapy angiogenics and for anti-GFR treatment. So we can see that these are various regimens which were used in these trials. So what we have done is we looked at the relative change in tumor size versus baseline. And we modeled this relative change as a function of time and logarithm of time in order to allow for curvilinear dependence of the relative change on time. In particular, for the control treatments, by the way, control treatments are listed in this table as the first in the row and the experimental as the second in the row. So for the control treatment, the assumed trajectory was a function of time and logarithm of time. And for the experimental treatment, we have modification of these coefficients of the intercept, time effect, and log time effect. So we can see that treatment effect can be expressed as any combination of these three coefficients. So we assume that the effect of treatment may possibly depend on time. But basically what we assume is that we have a trajectory of the relative change versus baseline depending on time, which is the trajectory can be different between the control and experimental arm. Now, because we use repeated measures of tumor size in time, or the relative change in time, we should account for the possible correlation between the measurements for individual patient. So we're including in these trajectories random time and or log time effects to account for this correlation. This is a technical step which is used in order to actually account for the possibility that measurements for the same patient can be correlated and therefore yield less information than just simply independent measurements or independent patients. So here's an example for one particular comparison. On the left-hand side, you see these spaghetti plots, as Everardo mentioned. So individual trajectories for the patients from the control group and from the experimental group. Well, one might see some, let's say, curvilinear shape, but it's of course difficult to make sense out of these plots. So this is the result of the fitting the model which I have just explained to these sets of observations. So you see two average profiles, one for the control group, which is the black curve, and one for the experimental group, which is the red curve. And as you can see, there seems to be a difference between these two profiles, indicating that the shift, the relative change versus baseline for the experimental treatment here is larger, so there's a larger tumor shrinkage as compared to the baseline, right? Now, based on this average profile, you can actually define, well, the depth of the nadir, the depth of response as Everardo mentioned. So this is the plot. So essentially what we look at this average plot and we see where the estimated trajectory achieves the minimum, okay? And for instance, in this case, for the control group, we would see that the nadir, the average of the nadir of the average trajectory is at minus 0.35. So this is like 35% reduction versus baseline. In the experimental group, the nadir is minus 0.39. So it's like about 39% reduction, so 4% reduction. We can also estimate the time to nadir. So you can see that the nadir value for the control group is reached at about 6.3 months, whereas the nadir at the experimental group is reached at 6.7 months, right? So now we could use these values and the differences expressing the treatment effect in terms of the depth of the nadir. So we could look at the effect of the treatment in terms of how on average the deep the nadir is in the two treatment groups and take the difference as a treatment effect. Or we could also look at another endpoint, which would be time to nadir, because well, one could say, well, that this time to nadir difference might also give an information regarding the efficacy of the treatment. If you would assume that, let's say, the rate of change for the two is sort of like comparable, then the earlier the nadir is reached, well, then it means that the less deep will be the nadir, and therefore the less the smaller the change versus baseline, and therefore the less efficacious treatment in bringing the shrinkage of the tumor. So what we've done is we have actually looked at these type of average profiles for all the treatment arms in all our trials, and we estimated treatment effects in terms of the depth of nadir or depth of response, and in terms of the time to nadir, and we looked at whether we could predict the treatment effect on survival from effect on these two endpoints. So, well, what I haven't included yet is the survival element. So for the relative change in tumor size, we use this model as I have just described, using time and log time as our time dimensions. We actually use proportional hazard model for overall survival, and in order to account for the association between the tumor size measurements and survival, we have included in this proportional hazard model random effects from this longitudinal part, tumor size part of the model, in order to account for this correlation. This is the methodology which exists since almost 15 years now, and it has been used to validate, for instance, also, PSA as a surrogate for survival in prostate cancer. So here's the set of comparisons for chemotherapy. These are survival curves. In some situations you see a clear difference in survival curves, in some situations you don't. And here on the right hand side you see the estimated average profiles. And again, in most of the cases there's very little difference between these profiles. In some cases you see a difference. Well, sometimes you see a clear nadir, sometimes you don't, okay? So, well, in most of the cases the nadir or time to nadir can be estimated. And here's the plot of the treatment effects on time to nadir, so the difference in time to nadir for the experimental control arm versus the log hazard ratio, and likewise the treatment effect in terms of the difference in nadir and log hazard ratio for overall survival. So what we can conclude from these plots is, and again, R squared is actually calculated here. So we see that there's quite a bit of spread of these points around the regression lines, which indicates that actually, again, we do not have a very precise, very strong relationship between these treatment effects. In fact, the relation is rather weak, and what we can say is that we have, in this case, like less than half of the variability on the treatment effect on survival explained by the variability in the difference in time to nadir, and even less so for the difference in nadir. Well, the question is, maybe for chemotherapy this is not, this is perhaps not working. Maybe it's different for the newer treatments. So here are the results for antiangiogenic comparisons. Again, survival curves and the longitudinal profiles. What is perhaps interesting is that to observe that there's a much clearer difference in these profiles as compared to the chemotherapy, and the difference seems to be in both rather in terms of the rate of the change in the relative change versus baseline. Now, if you look at the association between the treatment effects on time to nadir and difference in nadir versus the effect on survival, again, the association is not very strong. So, like for chemotherapy, although there seems to be a different dynamics of the tumor change or relative change, as I have explained it here, well, we don't see a clear association between the effects, treatment effects on these two endpoints and the effect on survival. So the third set of comparisons is the anti-GFR treatments. Now here again, what we could perhaps see is that the difference on the longitudinal profiles is rather in the shift. So it doesn't seem that the dynamics of the relative change is affected, but rather there seems to be like a constant shift of these profiles. But if we look at these comparisons and if we look at the estimated treatment effects, we again see that the association between the effects is relatively poor. So as I said, these are the tentative results, but again, if we look at the time to nadir or depth of nadir, these are like R-squared values for chemotherapy trials, for antiangiogenic trials, and for anti-GFR comparisons. We can see that nowhere more than 50% would be explained. It's actually less than half of the variability at most explained by the variability in treatment effects upon survival, which would be explained by the variability in treatment effects upon, sorry, I wrote response here, but obviously it's time to nadir or depth of nadir. So nadir of this endpoint seems to be valid surrogate at the trial level. So as Everardo said, there is interest in using this depth of response as perhaps a new candidate, but these results, at least in advanced colorectal cancer, do not seem to support the hopes that were let's say, formulated when looking or formulating this endpoint. Well, the last thing which we then could look at is, all right, so this is still like the question, okay, we look at the endpoint, and this is like for the treatment effect in terms of the of a single information regarding the depth of nadir or time to nadir, okay? But we could think about using the entire sequence of tumor size measurements as a surrogate, and we could actually look at the effect of the treatment, which, as we said, may depend on time, right? So we could actually try to look at the question, how about estimating the treatment effect, which may depend on time, and this treatment effect will be actually estimating using all the measurements without really necessarily reducing the longitudinal profile to depth of nadir or time to nadir, and maybe by using this whole bunch, this entire set of information, we could get an estimate of treatment effects on tumor size, which could then be used and to build a prediction model for the effect on survival. So the same, so using these models, this model which I have just described, we can actually look at the effect of the treatment as expressed by the shift in the profiles, by the effect on time, and by the effect on the logarithm of time. And this is the scatter plot matrix where we show the effect, so the shift in the intercept on the time scale and the log time scale, and this is the logarithm of hazard ratio over survival. And here we have like marginal associations between the shift time scale treatment effect and log time scale effect versus the logarithm of hazard ratio for overall survival. So the logarithm of hazard ratio is on the Y scale, and on the X scale we have, as I said, the intercept, the time effect, treatment effect, and the log time treatment effect. So for instance, from this picture, for chemotherapy comparisons, we do not seem to see much of an association, at least marginally, for each of these treatment effects. Now, if we take them jointly, we can build a regression model which would take this particular form, and we can again calculate R-squared. And again, the R-squared is very low. It's only about 0.16. So even if we use this information about the whole trajectory, which explains the treatment effect dependence on time, it doesn't seem to help us very much in predicting the treatment effect on the OS in chemotherapy trials. What about the antiangiogenics? Same story, scatter plot matrix. This is the plot for the treatment effects in terms of the intercept, time effect, and the log time effect. And again, there doesn't seem to be very much association. If we take it jointly, well, R-squared now jumps to 0.56. This is not a value we've seen previously, but still, it's only about the half of the variability, which is explained jointly by these three treatment effects. So although there seems to be in anti-angiogenics comparison, there seems to be more going on in terms of the association between these treatment effects, this whole set of treatment effects and the effect on survival, it's still not strong enough to allow us a good prediction. So the last set is anti-GFR subsets. Again, the plots which show not very much, and the model where now jointly it seems that things go better. So R-squared is equal to 0.63. The correlation underlying this might be seen to be 0.8. So it is about 2/3 of the variability. So one could say perhaps here there is some hope, but this is still not perhaps strong enough to have a very good surrogate. I mean, it might, of course, the precision of the prediction might be higher than in the previous cases which I have just shown, but I would say this R² should be much more close to 1 in order to say that we have a reasonable surrogate for the overall survival. And mind you, this is using the whole trajectory of all our measurements. So this is the most information we can squeeze for the tumor size measurements that there is there, right? So in a sense, I would say that this is like a maximum that we can squeeze out in terms of the potential endpoint, tumor size based endpoint that we could use in order to infer the treatment effect. So, well, these are the, as I said, the results which we have just worked on using this ARCAD database, and the tentative results actually lead to the concluding remarks that Everardo will gladly present.
Okay, thank you, Tomasz. So we hope to have at least shown you in general terms that tumor shrinkage is a good indicator of treatment effect in individual patients, and that's what has been found in most of the analyses so far. On the other hand, as surrogates for overall survival, we've yet to see the role of these endpoints, especially the novel ones. We already knew that response rate itself, at least for the tumor types for which it has been assessed, is not a very strong surrogate for OS. And for these novel endpoints, we have less information for the time being, but it appears that they are also not very good surrogates. And it appears also, based on limited information, that there seems to be no preference regarding how tumor size is assessed, whether you look at it as a continuous variable or as a dichotomous variable. At least for the sake of surrogacy, it seems that that doesn't matter. So this is what we had to present to you today. We hope you have submitted questions. If not, please do so. And I will hand it over to Edward again, who will take care of the Q&A session. Thank you very much.
Thank you so much, Dr. Saad, and thank you, Dr. Berzykowski, for that insightful presentation. Now, I would like to invite our audience to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. I've already received some questions here, and so we'll get started with those. So question 1 here asks if RR is not a surrogate for OS, then why is it used as endpoint in phase 2?
Yeah, I guess I can tackle that one. Thank you for sending this question. Quite frankly, I don't know the answer. I guess it has been used for historical reasons. Especially in phase 2 trials, it's seen as a good screening for the activity of agents. It's true that in phase 2 trials you can not always afford to wait for the long-term outcomes, such as progression-free survival and overall survival. So I guess for historical reasons it has been used. Most of the times it has not been proven to be a good surrogate in solid tumors, especially for OS, and I think we'll continue to use it because it serves us well, I think. So essentially it's, I would say for historical reasons mostly. Tomasz, do you have anything to add to that?
Well, I mean, I understand the hardest point clinically it's useful measure, but I mean, in a sense,
sorry to chime in, Thomas. I think you might be muted on your end. We're having a little bit difficulty hearing you.
Yeah, no, I didn't unmute myself.
That's much better.
I
Thank
think
you.
I'm now on. Yeah, what I was trying to say is that, well, indeed the question is correct, and although Everardo points to the clinical view, I mean the question I would pose is, okay, fine, indeed, I mean, if you see that there's no link, the question is, I mean, if we promote drugs, based on response towards the phase 3, don't we then risk what we actually observed, that many phase 3 trials failed, right? And indeed, I mean, well, from looking from this point of view, my point would be, well, we should look for something better, but to look for something better, we have to understand the mechanism of treatment, I guess. So nowadays with the targeted therapies, well, maybe it's easier to come up with biomarkers which would help us in deciding whether the treatment is doing this job properly. But in the past, I understand, tumor response was the most tangible thing to evaluate the activity of the treatment. So, well, this is what Everardo said. I guess that for historical reasons, in the era where we didn't understand the mechanism of treatment very much, well, this is what has been done.
Great, thank you both for that answer. Another question from another attendee here. Can the sample size for phase 2 trials let me repeat that. Can the sample size for phase 2 trials be estimated using tumor shrinkage as a continuous variable? And if so, why isn't this done?
Yeah, let me start again, Everardo here. Yeah, of course, yeah, it can be used. Arguably, it would provide for more power, depending, of course, on the variability of the relative change or however you measure it. But again, probably for historical reasons, it hasn't been used, which makes it difficult for anyone designing a new study because you don't have the published results from previous trials. So that's one of the problems. Of course, you can say you have some pilot data, or you can try to sort of derive those data from publications. But also you would create a problem of interpreting the results, because since most of the literature is based on the response rate, when you look at relative change, it's not very clear how you go from one to the other. So it's true that in the future, I guess we'll see more and more of responses being analyzed as a continuous variable, but for the time being, it hasn't been used a lot.
Well, maybe I will also chip in quickly here. One of the points which might be also mentioned is that, well, from a practical point of view, currently the question is, what is done usually is that we measure let's say the tumor volume is measured on the so-called target regions, right? So you can have also non-target lesions and lesions which are newly appearing, right? And then the question is, okay, fine, if we would do the sample size calculation based on the tumor size, well, this is also the point, what tumor size we have on mind, in a sense, right? Is this only the target lesions which we would, use as they are currently used, or should we? So this is also the question, as I think what Everardo points to is that this endpoint hasn't been used much, so there's not that much, let's say, information, and both from the point of view of definition and from the point of view of interpretation, it's probably not very easy, or not, well, currently it's not very often used, and for this reason it's then difficult to promote it as an endpoint in new trials.
Okay, great. Thank you for that. This question here asks, what is the R-square for PFS HR versus OS HR? I don't think it is reasonable to expect the R-square for tumor shrinkage versus OS HR to be close to 1.
Well, maybe it is not worth expecting, but this is what you would like to have for being able to predict, right? So, well, what one says in this concept of surrogacy is that you want to predict, let's say, hazard ratio for overall survival from the hazard ratio on PFS. It means that you need a prediction model, right? If you use linear regression, well, you have to essentially have a high R squared in order to have a reasonable prediction. Now, obviously one could debate what is the reasonable prediction, right? And this is not something that has been settled yet, right? But in a sense, the idea is that, you know, you should have high R squared in order to be able to meaningfully say with, and meaningfully means with narrow prediction limits, that your effect on overall survival as expressed by hazard ratio will be this with relatively narrow confidence limits, right? And yes, if you say that it's not reasonable to expect that the R squared for the association between the hazard ratios on OS on PFS will be high, well then essentially it's like answering the question about the surrogacy. This means that the PFS will not be a good surrogate for OS. And well, obviously one can say, well, from my experience, I don't expect that, but it's also worthwhile to check that. And well, in other cases, for other endpoints, and not, I mean, actually, in some situations, well, it seems that this association is strong, and actually for advanced colorectal cancer, at least for the 5FU based regimens, the association was pretty strong, okay? So in advanced cancer, advanced colorectal cancer, it could be seen that the PFS, the hazard ratios on PFS and OS are quite closely associated, but in other situations it's not the case. So it really depends on the, it seems to depend on the disease, and in general, yes, the intuition may be right, but well, this is exactly the criterion you would like to check empirically before concluding that, you know, you cannot use PFS as surrogate for OS.
Great, thank you for clarifying that, Dr. Berzykowski. Another question here, this audience member asks, with respect to specificity of tumors in different organs, can the model be standardized across various tumor types? Example for tumor size in lungs or the liver or the brain, which has progressed within the same amount of time?
I guess this one might be for me. I'm not exactly, hold on, I'm not exactly sure what would be the standardization. I mean, in a sense, what you could say is that you could use a similar model where with the, let's say, what is important, and I understand that in the terms of the model, what is important is the trajectory over time. So whether the profile can be estimated, right, in more or less the same way. And well, that's a question, well, in principle it's the question about choice of parametric function. Now in our modeling we use this time and log time, a similar type of well, so this allows you to capture, for instance, this slowly, you know, going down rapidly to the nadir and then slowly going up. I mean, in principle also, let's say, a little bit different dynamics can be captured by this combination. So now one could say that, well, in this respect, this may be, let's say, in quotation marks, standard approach which you could apply to other situations, but obviously, I mean, if there are some a little bit more like abrupt behaviors, let's say like almost like quadratic type of dependence, well, this model may be a little bit maybe misspecified. So it's just a question about the parametric form which you might want to use to model the dependence on time. And well, to which extent the same parametric form, as I say, time plus log time, may be used in other cancers. Well, honestly, I'm not that expert in tumor dynamics in other diseases, so it would have to be seen against the empirical data to decide whether the type of dynamics is similar. So standardization in terms of using the same type of parametric function, in principle, you could use the same parametric form. The question indeed is whether this form is flexible enough to capture, well, the behavior in different diseases. Well, that I'm not sure whether I can answer this particular question, whether, you know, in some diseases the dynamics may be more rapid than for instance, in advanced colorectal cancer.
All right, thank you both for those answers. We have reached the end of the question and answer portion of this webinar. If we weren't able to attend to your questions today, the team at IDDI may try to follow up with you after the webinar. If you have further questions, please direct them to the email address showing on your screen right now. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. I am just sending everyone a link in the chat right now. You can say thanks to our speakers via Twitter using that link, so I encourage you to join us in thanking Dr. Berzikowski and Dr. Saad for the wonderful presentation. Thank you both.
Thank you, Edward.
We hope you found this conference informative. Have a great day, everyone.